Welcome students, I am Bittu Singh from AISSMS College of Hotel Management and Catering Technology Pune. I will be introducing you to this session of Environmental Science 1 under the Bridge course. The topic for today's session will be Conservation of Biodiversity, In situ and Ex situ Conservation of Biodiversity Part 2. In our previous session, we discussed about importance of biodiversity, conservation and various methods of in situ conservation in detail. Now, in this session, we will discuss about ex situ conservation method in detail and differentiation between in situ and ex situ methods. Now, let us take a look at the objective of this session. In this session, students will be able to understand definition, key points, methods, advantages, and limitations of ex situ biodiversity conservation, and they will be able to differentiate between in situ and ex situ methods. Now let us see what is ex situ conservation method. Ex situ biodiversity conservation refers to one of the approaches of conserving biodiversity that ensures the survival and sustainability of endangered plants and animal species outside their native habitat. It provides an artificial environment for diverse plants and animals similar to their actual growth conditions. It is also termed as off-site conservation. Human activities and some natural disasters are the factors responsible for global decline in biodiversity. Thus, ex situ conservation is one of the approaches to cope up with the consequences of declining biodiversity. Ex situ biodiversity conservation primarily targets endangered species, species with local importance, ethnobotanical interest, and taxonomically isolated species. Let us see here definition of ex situ biodiversity conservation. It refers to the method of conserving genetic species and ecosystem biodiversity by artificially providing habitat close to their natural environment. Zoological gardens, botanical gardens, aquariums, and gene banks are ex situ conservation methods. It is the conservation of biodiversity outside a species' natural geographical range. Besides conservation, it also ensures restoration and reintroduction of species by preserving the genetic and reproductive material of desired species that are about to extinct from the population. Let us understand here some of the key points related to ex situ conservation. First is transfer of threatened, endangered and extinct species. This involves transferring threatened, endangered and extinct species away from their native environment. Second, artificially created environments. It targets plants and animal population. Ex situ conservation provides artificially created environment or conditions alike to their actual habitat to the plant and animal population. Third is cryopreservation. It increases the genes, seeds, sperms, ovules, etc. viability through cryopreservation. Fourth, survival of the threatened species. Survival of the threatened species is the primary approach of ex situ conservation. Fifth is maintenance of genetic diversity. It also targets the maintenance of genetic diversity. Sixth key point of ex situ biodiversity conservation is preservation process. Ex situ conservation of plants is mainly established to preserve seeds, pollens, and ovules. Plant tissue culture and plant breeding are also part of ex situ conservation, through which plants are produced in vitro. Ex situ conservation of animals is established to preserve semen, ova, and DNA. Captive breeding and embryo culture are also the techniques that allow the reintroduction of the species. It conserves genetic resources, wild species, and cultivated species. Next key point is knowledge of the genetic structure, regeneration method, and maintenance. Knowledge of the genetic structure of target species, regeneration methods, and maintenance of gene pool become necessary to establish ex situ conservation. These are the methods of ex situ conservation. Here you can see zoological gardens, botanical gardens, gene bank, and aquarium are some of the methods of ex situ conservation. We will discuss these methods in detail in further slides. Let us begin with zoological garden. Zoo or animal park refers to the place that displays animals living in captivity away from their natural environment. Zoological gardens provide food, shelter, and medicine to a wide variety of animals and birds. There are more than 164 zoos in India. Zoos provide a confined space to the animals through small cages, paddocks, and fenced-in fields. Programs on animals' well-being, captive breeding, environmental education, and exhibitions are also provided in the zoos. Captive breeding provides an important tool to prevent the extinction of animal species. Its primary goal is to conserve the maximum genetic variability within species. Captive breeding reduces mating between closely related individuals in order to minimize inbreeding and loss of genetic variation. Curators in the zoological gardens are exceptionally trained in the animal acquisition and transfer. There are keepers who perform routine checkups of the animals. There are veterinarians focus on preventive measures against contagious diseases, accidental injuries, and autopsies. Trained volunteers guide visitors and also monitor their actions in the zoological gardens. Let us see another method of ex situ conservation, that is botanical garden. It displays a variety of rare and exotic plant species. The botanical garden provides an essential tool for education and research purposes. It allows the growth of flowers, fruits, and vegetables by providing similar growth conditions and nutritional requirements. Plantings are scientifically ordered based on their phylogeny and taxonomy. It also includes nurseries and greenhouses to propagate and cultivate exotic plants. It helps in studying the tropical and temperate ecosystem. The botanical garden maintains the sustainability of abandoned ornamental plants. The botanical gardens conserve endangered and rare plant species. It looks aesthetic to the visitors. Let us see here another method of ex situ conservation, that is gene bank. It functions as a biorepository. The gene bank preserves the animal's genetic material as well as plants' structures with regeneration capacity. It involves the freezing of animals' sperms and eggs in zoological freezers. It also freezes the plants' cuttings and seed stalks. Coral fragments can also be preserved in water tanks by providing a controlled environment. Thus, the gene banks serve as a center for keeping genetic and reproductive material. It uses the cryopreservation method to store the reproductive and genetic content of the plant and animals. There are many types of gene bank which are as follows. First is seed bank. It stores seeds to conserve genetic diversity. Seed bank or seed libraries has a primary approach to increase yield, disease resistance and nutritional quality of plants. Here, cryopreservation preserves the seed's viability for the long term. We can introduce genetically modified methods through seed bank. Next type of gene bank is in vitro bank. It involves the preservation of buds, meristematic cells and tissues in a semi-solid or liquid nutrient medium. It is a kind of slow growth conservation method used prevalently to conserve seedless plants. Next type of gene bank is cryobank. It preserves seeds and embryo through cryopreservation at 196 degrees Celsius. Cryobank targets to conserve extinct species. It is also helpful for the preservation of animal genetic resources. Another type of gene bank is pollen bank. It stores the pollen grains in liquid nitrogen. Pollen banks play a pivotal role in the crossbreeding. Next type of gene bank is field gene bank. It is the method of planting plants in an artificially created environment. We can conserve the germplasm of essential plants through this method. It needs more land area, soil, and is vulnerable to pests and diseases. Next method of ex situ conservation is aquarium. It seems like a vivarium that varies in size. Aquarium houses aquatic plants and animals. The plants in the aquarium give enough oxygen to water bodies within the aquarium. We can keep small aquariums at home. Large public aquariums house fishes, otter and turtles. Glass or acrylic typically frame the body of the aquarium. Fish tanks are the cuboidal aquaria and fish bowls are the oval or bowl-shaped aquaria. Aquarium requires high maintenance and an aquarist must be skilled in operating the components of aquarium. The filtration system, air diffuser, pump and thermostat are the components of an aquarium. These were the methods of ex situ conservation. Now let us see advantages of ex situ biodiversity conservation. First, it requires less area. Second, threatened species are protected from all adverse factors like predation and poaching. Third, under human supervision, plant and animal health can be monitored and medical assistance can be given as required. Fourth, any disaster and disease management becomes easy in ex situ biodiversity conservation as it only conserves the small population. Fifth, we could measure the genetic diversity of the population. Sixth, we can conduct selective breeding programs for the animals and plants to increase their numbers if endangered. Seventh, it is also helpful for the educational purpose as researchers and educators may study the reproductive physiology, lifestyle and ecology of endangered species up close. Ex situ collection sites could raise funds for conservation efforts. Let us now see some of the limitations of ex situ biodiversity conservation. First is ex situ biodiversity conservation is applicable only to conserve the small population. Second, it requires more expense to conserve the threatened plants and animal species as they require special care and attention as well as basic needs like food, shelter and medicine. Ex situ conservation involves several expensive methods like cryopreservation and tissue culture to preserve the genetic and reproductive material. It provides less mobility to animal population due to the restricted or small space. Animals are vulnerable to a wide range of diseases due to translocation from their natural habitat. Animal may not survive reintroduction into the wild habitat or they can face difficulties with acceptance by the existing wild species. Nutritional issues may also arise in the ex situ biodiversity conservation method. These were the limitations of ex situ biodiversity conservation. Now let us see difference between in situ and ex situ biodiversity conservation for our better understanding. Let us see in situ conservation. In situ conservation means conserving all the living species, especially all the wild and endangered species in their natural habitat and environment. And ex situ conservation means conserving all the living species in the man-made or artful habitat that are similar to their natural living habitats. Here in this table, we will be able to see some of the points that differentiate between in situ conservation and ex situ conservation method. So first is in situ conservation. This is on-site conservation, and ex situ conservation is off-site conservation. In situ conservation involves the natural habitat of organism, and ex situ conservation involves the man-made habitat. In situ conservation is appropriate for animal found abundantly, and in situ conservation method is appropriate for animal found abundantly, and ex situ conservation method is appropriate for animals which are not found abundantly. In situ is not suitable in case of rapid decline in number of the species due to any factor, and ex situ is the best option in case of rapid decline in the number of a species due to any factor. In situ can be done for wildlife and livestock conservation, and ex situ conservation can be done to conserve crops and their wild relatives. In situ conservation, designation, management and monitoring of the target species is involved, and in ex situ conservation, sampling, storage and transfer of target species from their natural habitat to the man-made habitat is done. In situ helps in maintaining the naturally ongoing processes of evolution and adaptation within the natural habitat of all the species, whereas ex situ conservation separates the animals from their naturally ongoing processes of evolution and adaptation. And examples of in situ conservation method is national parks, wildlife sanctuaries, biosphere reserves. Example of ex situ conservation is zoo, cryopreservation, DNA bank, aquariums, botanical gardens, etc. So here we were able to understand difference between in situ and ex situ conservation method. We also discussed about various methods of ex situ conservation in detail. Now let us conclude this session by understanding this. Biodiversity conservation is necessary to maintain the ecological balance, continue natural processes and support the ecosystem. In situ and ex situ conservation are two crucial approaches to preserving biodiversity, while in situ conservation focuses on protecting organisms within their natural habitat, whereas ex situ conservation involves safeguarding them in a controlled environment, and it has a practical approach to protect rare and endangered species. Both methods aim to safeguard species but through different means, highlighting the importance of employing diverse strategies to ensure the survival of Earth's diverse ecosystems and species. Thank you.